We can start. Hello, everybody. And we want to give you a brief summary of our recent paper. I'm Guillermo Oliver, Director of the Center for Vascular Developmental Biology at Northwestern, and this is my friend and colleague.
Hi, everyone. My name is Ed Thorpe. I'm an associate professor at Northwestern University, collaborating here with Guillermo Oliver.
And we want to give you a brief overview of our work and basically how this collaboration started. And for that, we will share a short PowerPoint that we prepared for this purpose. So let's start by saying that this work was performed by the two co-first authors, Chris Clinton, postdoc in Ed's lab, and Wan Shuma, postdoc in the Oliver's lab. And basically this initiated a little bit based on relatively recent data that has demonstrated the important function of cardiac lymphatics in the resolution of inflammation following MI. Basically has been shown that there is increased lymphoangiogenesis after cardiac injury, and this is basically promoted by a growth factor called VEGF-C, and as is shown in this diagram, the lymphatics grow in the infarcted area, as you guys can see here. My lab has been interested in lymphatics and lymphatic development and lymphatic function for a long time, and we somehow enter into the role of lymphatics in cardiac development and cardiac function a little bit by accident, and was somehow this paper published a couple of years ago by my lab somehow highlighted this role of lymphatics in cardiac repair. We showed that a factor called renin produced by lymphatics help to repair the injured heart. And the question was always, where does VEGF-C comes from? And that's where the interest with Ed's lab comes from, and that was this exciting collaboration came forward. Ed will continue with this explanation.
Yeah, thanks, Guillermo. So after a heart attack or myocardial infarction, the clinical therapy is something known as percutaneous coronary intervention. And this is ultimately a protective treatment that activates cardioprotective response by the innate immune system, including macrophages, which help to contain the infarct and help repair the heart. Unfortunately, in some cases, this can also lead to collateral ischemia reperfusion injury, where the macrophages can do also harm to the heart. And so really, the opportunity here, as from a basic science perspective, is to understand the dichotomy of the good things that macrophages do versus the bad things they do. One of the good things that macrophages do during tissue injury is to promote this process called efferocytosis. This is the clearance of apoptotic cells, and it's not just the clearance and the removal of dead cells, but also activates macrophage reprogramming and secretion of factors that can affect cardiac repair. So one of the first questions that we asked together in collaboration in this publication was whether or not efferocytosis might be a trigger for growth factors, including VEGF-C. This was done very simply in a tissue culture dish where we co-incubated macrophages with apoptotic cells. And what we discovered was that macrophages could produce or induce the transcription of VEGF-C mRNA after exposure to apoptotic cells. This is shown in panel A. We also found that this process of induction of VEGF-C was dependent on a known phagocytosis receptor called CD36 in panel F. And furthermore, if we went into mice in which we had induced experimental myocardial infarction and sorted out CD64 macrophages which had eaten cardiac antigen, we found that those macrophages which were eating more cardiac antigen in panel C also expressed the most VEGF-C. So subsequently, and in close collaboration with Dr. Oliver's lab, we leveraged these mice in which VEGF-C was specifically deleted in a myeloid compartment through construct lysem-Cre. And so the question here was whether or not, or where the source of VEGF-C after MI might be. And as you can see in the depiction here in experimental mice in the top panel, in essentially control or wild-type mice at day 28, there's significant evidence of LIVE-1 positive cardiac lymphangiogenesis after myocardial infarction. In contrast, in the lower panel of A, you can see that there's a significant reduction in this expansion of these vessels. This was functionally correlated by measuring the echocardiography or utilizing echocardiography after MI, and you can see those mice which had deficiency of VEGF-C in macrophage-like cells had a reduced ejection fraction in panel 5A. It's also been shown that in some cases, macrophages can express receptors for VEGF-C, and so we asked the question, does VEGF-C potentially regulate the production of cytokines which are critical for cardiac repair? And in culture, we added VEGF-C to macrophages which were inflamed through activation of toll-like receptors, and we found that VEGF-C could suppress the production of pro-inflammatory cytokines TNF-alpha and IL-12. So taken together, we propose a working model in which during injury, for example, when you have exposure to dying cells, macrophages utilize their phagocytosis receptors, such as CD36, to turn on a signal transduction pathway that leads to the production and secretion of VEGF-C. This is important in inducing a cardiac lymphangiogenesis response, which affects cardiac repair and improvement of cardiac function. Also, interestingly enough, VEGF-C may also feed back on the macrophages to directly regulate cytokine production.
So, yes, we obviously want to thank everybody that was involved in this work, obviously our funding agencies, but we believe in summary that this work provides a nice link, first supporting these findings and the idea that lymphatics or lymphangiogenesis promoted by VEGF-C indeed helps, improves cardiac repair after MI. But now I think we have evidence also to say that one of the major inducers of sources of VEGF-C are, as Ed was indicating, was macrophages that accumulate around the injury areas. So thank you for your attention. Have a good day.